Welcome. Today, we are bringing you a deep dive into an innovation that sits right at the intersection of high-level mathematics, massive data architecture, and ultimately human compassion.
Yeah, it's a really profound topic today.
It really is. We're exploring the core concepts from an analysis titled Synthetic Control Arms, the Ethical Revolution in Clinical Trials. And the mission of our deep dive today is to explore a pretty massive shift in medical research. We're looking at the attempt to actually replace the placebo and whether doing that could dramatically accelerate cures for the most vulnerable patients out there.
Right. It's a concept that fundamentally challenges the mechanics of how we have evaluated medicine for, gosh, nearly a century now. We are looking at a methodological shift that doesn't just introduce a new statistical tool, but it forces a complete reevaluation of how we quantify human time in the pursuit of scientific rigor.
Absolutely.
The implications for you, your family, and the broader healthcare ecosystem are just vast.
They really are. And to ground this in reality, I want you to imagine a very specific scenario. Picture yourself as the parent of a 5-year-old child who has recently been diagnosed with an ultra-rare neurodegenerative condition.
That's a devastating diagnosis.
Beyond devastating, the medical consensus is bleak, there's no cure, and standard treatments are essentially non-existent. You spend your days just watching your child slowly lose developmental milestones. But then, a breakthrough happens. A biotech firm announces a phase 3 clinical trial for a novel gene therapy that has shown staggering promise in early studies.
It's the exact miracle you've been hoping for.
Exactly. You apply, and your child is accepted.
But that acceptance comes with the stark reality of modern clinical trial design. The consent form outlines the protocol. It is a randomized, double-blind, placebo-controlled trial.
Okay, let's unpack this. We all understand the basic mechanics of a randomized trial, right? You flip a coin, half the participants get the experimental therapy, and the other half get a sham procedure to serve as the baseline.
Right, the control group.
Yeah. But for this parent, that 50-50 coin flip means risking placement in that control arm. And if that happens, the child doesn't just miss out on the drug. They lose the critical, incredibly narrow biological window where the therapy might have actually halted the disease.
That's the real tragedy of it.
By the time the trial concludes, the degeneration could be completely irreversible.
The clinical community actually refers to this tension as the control arm dilemma because we all know why the randomized controlled trial, the RCT, has been the gold standard since the mid-20th century.
Because it works.
Because it works beautifully from a mathematical standpoint. Physical randomization is mathematically elegant. By taking a group of, say, 200 patients and randomly splitting them, you effectively balance out all the unknown unknowns.
You account for the placebo effect.
Exactly. You account for the placebo effect, and you isolate the natural progression of the disease. Any difference at the end of the trial can be confidently attributed to the drug itself.
And the ethical bedrock supporting that mathematical elegance is the concept of equipoise.
Equipoise, yeah.
It's the idea that placing people in a placebo group is only justifiable when the medical community genuinely does not know whether the new drug is better, worse, or neutral compared to doing nothing.
Right. If you know the drug works, you can't ethically withhold it.
Precisely. But the central argument we are exploring today is that this concept of equipoise is fracturing. It's braiding under the weight of modern medical breakthroughs.
If we connect this to the bigger picture, the sheer velocity of modern medicine is outpacing the structural capacity of our traditional trial designs. The researchers in the analysis point to 4 distinct compounding pressures that are creating a breaking point in how we conduct these studies.
Let's examine those pressures, starting with the first one, recruitment and precision medicine. We've moved far beyond treating broad categories of illness. We aren't just recruiting lung cancer patients anymore.
Far from it. An oncologist today isn't just looking for lung cancer. They're looking for a patient with non-small cell lung cancer, featuring a specific ALK genetic mutation, who has already developed resistance to 2 prior lines of targeted therapy.
Wow, that's incredibly specific.
It gets even more granular. They also need to exhibit a specific protein expression profile. We are slicing the patient population into these molecularly defined micro-cohorts.
So finding these people is like finding a needle in a haystack.
Exactly.
Yeah.
In the realm of rare genetic diseases, there might be fewer than 500 patients globally who share a specific profile. Finding 100 of them for a trial is a logistical nightmare.
I can imagine.
And telling half of those incredibly rare individuals that they must serve as the control group and receive a placebo, it's practically impossible from a recruitment standpoint.
Which feeds directly into the second pressure, which is time and cost. Developing a new drug currently takes an average of 10 to 12 years.
And costs upwards of $2 billion.
$2 billion, that is staggering.
Yeah.
And a massive portion of that capital and that time is entirely consumed by the logistical friction of finding, enrolling, and then retaining that control arm over years.
Then there is the third pressure, known as the generalizability gap.
Yeah.
This represents a structural flaw in the utility of traditional RCTs. Well, to maintain that mathematical purity we talked about, traditional trials require a highly controlled environment. They predominantly take place in elite research hospitals, the ivory towers.
Right.
And they routinely exclude the elderly, patients with complex comorbidities like someone with cancer who also happens to have diabetes and hypertension.
Which is a lot of people.
A huge portion of the population. They also exclude patients from diverse socioeconomic backgrounds who simply cannot easily travel to a specialized clinic every single week.
So they are essentially testing these drugs on clinical athletes.
Clinical athletes, that's a perfect way to put it. It creates a pristine data set for the researchers, but it leaves doctors guessing how the drug will actually perform in the messy, complicated real world.
The real world where patients might occasionally miss a dose or they're taking conflicting medications.
Precisely. Which leads to the fourth and final pressure, the ethical breaking point.
This one hits hard.
It does. Consider oncology. If an early phase 2 trial for a targeted cancer therapy demonstrates an 80% tumor response rate, the traditional requirement to then run a massive phase 3 trial with a placebo group,
It just feels wrong.
it no longer feels like a pursuit of scientific rigor. To the patients and to many of the clinicians actually treating them, it feels like an artificial barrier to survival.
So to summarize where we're at, we have highly targeted drugs for tiny populations, taking over a decade to develop, costing billions, tested on unrealistic patient profiles, all while agonizing the exact patients we were trying to save.
That's the grim reality of the bottleneck.
So how do we fix it? How do we reconstruct the mathematics of a control group without using real human beings as placeholders?
This is where the synthetic control arm, or SCA, enters the equation.
Here's where it gets really interesting. An SCA is essentially a virtual control group built entirely from real-world data. We call it RWD. This involves harvesting the digital footprint you already leave behind in the healthcare system. We're talking about everything from your electronic health records and insurance claims to disease registries.
Even data streaming from wearable diagnostic devices.
Exactly. So instead of recruiting 50 real people to take a placebo today, researchers construct a control arm out of the historical data of people who have already experienced the standard progression of the disease in the real world.
But to achieve the mathematical rigor of a traditional RCT, you cannot simply grab a random batch of historical patient files. You need precision. The foundational mechanism here is patient matching, which relies on some truly formidable statistical engineering.
Okay, let's get into the math.
Two of the primary techniques utilized are propensity score matching and inverse probability weighting.
Let's demystify those terms for a moment. If I understand the analogy used in the source analysis correctly, propensity score matching functions somewhat like a highly sophisticated dating app algorithm. But instead of personal interests, it's optimized for clinical vectors.
That is a very effective way to visualize it. Imagine you have a real patient enrolled in your active trial. Let's say a 45-year-old woman with stage 3 breast cancer, possessing very specific tumor biomarkers.
Okay.
To build her counterpart in the synthetic control arm, the algorithm scours massive data sets of real-world data to find her digital twins.
Digital twins, I love that term.
It generates a probability score to find historical patients who match her exact clinical profile. They are matched on age, sex, disease severity, prior treatment history, and specific comorbidities like diabetes.
And inverse probability weighting. That sounds like the algorithm is actively manipulating the data to fix imbalances.
It is essentially a balancing scale.
Yeah.
Let's say a certain demographic, for example, elderly patients with a specific cardiac condition, is underrepresented in the historical data compared to your active trial group.
You have a mismatch.
Right. The algorithm assigns a heavier mathematical weight to the outcomes of those few elderly historical patients. It ensures the virtual control arm perfectly mirrors the demographic and clinical makeup of the real trial arm.
It neutralizes the variables that could skew the results.
Exactly.
So what does this all mean?
It means we can theoretically bypass the need for a concurrent placebo group entirely.
Which is huge.
It's revolutionary. You build this meticulously matched synthetic cohort, and you analyze their historical medical records to determine their trajectory. How long did they survive? How quickly did their disease progress under the standard care of their time?
And then you just compare those historical baselines against the outcomes of your active trial patient.
You got it.
The downstream effects of that are massive. Every single patient who enrolls in the trial receives the experimental, potentially life-saving treatment. The 50-50 coin flip is eliminated.
Go on.
And because you are bypassing the years-long process of hunting down patients willing to risk taking a placebo, the timeline to get a drug approved and into pharmacies shrinks dramatically. It cuts off months, sometimes years.
What's fascinating here is how rapidly this methodology is moving from statistical theory into active clinical application. It's happening right now. The analysis details 3 critical arenas where synthetic control arms are already reshaping research, beginning with rare diseases.
This brings us to the N of 1 reality. For those unfamiliar, an N of 1 scenario happens when a genetic mutation is so unique that a clinical trial might literally consist of a single patient.
Or perhaps a tiny handful of people globally. A perfect example is spinal muscular atrophy, or certain ultra-rare metabolic enzyme deficiencies.
Right.
If there are only 30 children in an entire country with a specific condition, running a traditional 200-person randomized trial is a mathematical impossibility.
You just don't have the bodies.
Exactly. By leveraging an SCA, researchers can administer the experimental therapy to all 30 children. They then measure their physiological progress against a synthetic control arm built from natural history registries.
Data detailing exactly how the disease historically paralyzed or impacted children before the new drug existed.
Yes. For these incredibly rare conditions, the synthetic approach isn't just a shortcut. It is the only viable mechanism for scientific validation. We literally could not test these drugs otherwise.
Oncology is another major frontier for this, driven by the sheer speed of innovation. The standard of care in cancer treatment evolves so rapidly now.
It changes almost yearly.
Right. So if you initiate a traditional trial today, the standard treatment you are using in your control arm might be completely obsolete by the time the trial concludes 5 years from now.
This velocity is exactly why SCAs are facilitating single-arm trials in oncology. An investigative team can administer a novel immunotherapy to a cohort of patients and compare their tumor shrinkage against a virtual cohort of patients who received what was considered the best available treatment 2 years prior.
It ensures that no patient in the current trial is relegated to receiving outdated, inferior care simply to satisfy a control requirement.
Exactly. The third crucial application highlighted in the research involved post-market studies, which are effectively known as Phase 4 trials. This circles back to that generalizability gap we talked about earlier.
Right, the clinical athletes versus the real world.
Once a drug clears the FDA and hits the market, doctors need to know how it actually affects the populations who were excluded from the original pristine trials,
Like pregnant women.
or elderly patients who are already taking 6 other medications. Historically, answering those questions required launching massive, prohibitively expensive follow-up trials.
But now,
Today, utilizing SCAs, pharmaceutical companies can monitor the real-world efficacy and safety of a drug in those specific populations by aggregating their electronic health records as they are naturally prescribed the medication by their primary care doctors.
all of this sounds incredibly promising. But I want to channel some healthy skepticism here, put on my learner hat.
Please do.
If we can achieve all of this simply by manipulating data, why hasn't the entire pharmaceutical industry abandoned the old RCT model overnight? If it sounds too good to be true, where is the catch?
This raises an important question, and it is the primary reason regulatory bodies and statisticians maintain a stance of aggressive caution. The undisputed monster under the bed when constructing a synthetic control arm is bias and confounding.
Let's dig into that, because earlier we discussed how propensity score matching handles the clinical variables. If the matching is really that rigorous, where's the bias coming from?
The vulnerability lies in the fact that an algorithm can only match patients based on the variables explicitly recorded in the dataset.
Okay.
In a physical randomized controlled trial, the act of flipping that coin and physically randomizing patients magically balances out the unknown unknowns, factors the researchers didn't even know they should be measuring.
Ah, I see. So if a historical patient lived 5 years longer, but the reason they survived was because of a specific dietary habit or an obscure genetic marker that doctors weren't even testing for a decade ago.
The algorithm is entirely blind to it. It can't see what isn't typed into the computer. If that unrecorded factor was the true driver of their survival, It creates a hidden bias in the synthetic arm.
which skews everything.
It could artificially inflate or deflate the historical baseline, making the new experimental drug look falsely miraculous or falsely ineffective.
That structural vulnerability is compounded by the issue of data quality, right? We call it real-world data, but the reality of medical documentation is that it is incredibly noisy.
Very noisy.
This data isn't being entered by dedicated clinical trial coordinators whose sole objective is immaculate data entry.
No, it is being entered by exhausted nurses and physicians during chaotic shifts in emergency departments.
Exactly.
It is plagued by missing values, typographical errors in coding, and highly subjective interpretations of what constitutes a patient, quote-unquote, improving. The foundational rule of data science applies here: garbage in, garbage out.
If the electronic health records forming your synthetic arm are fundamentally flawed, the resulting comparative analysis is scientifically void. So how does a biotech company convince a highly skeptical scientific community that their virtual control arm is actually robust?
They do it through a rigorous process of validation. This often involves what are essentially mock trials or target trial emulations.
How does that work?
To build trust, a research team will take a highly successful, fully completed randomized controlled trial from, say, 10 years ago. They will digitally amputate the real control arm from that historical study, throw it in the trash.
Okay.
Then, using real-world data from that exact same time period, they will attempt to build a synthetic control arm from scratch.
Oh, I get it. It's a retrospective stress test.
Exactly.
Yeah.
If they run the math on the synthetic group and the resulting survival curves perfectly overlap with the survival curves of the real people who took the placebo 10 years ago, it proves the methodology works.
It demonstrates that the statistical engineering is capable of filtering through the noise of real-world data to arrive at the objective truth.
And regulatory agencies are scrutinizing this validation process intensely. Between 2023 and 2024, both the FDA in the United States and the EMA in Europe issued landmark guidance frameworks regarding the integration of real-world data.
So they aren't shutting the door on it. They're just demanding that sponsors show their work.
Yes, regulators require exhaustive proof that the data utilized is fit for purpose. They expect total transparency regarding the algorithms used for patient matching, the protocols for cleaning the noisy data, and above all, the cryptographic measures employed to ensure patient privacy is protected at every single layer of the computational process.
It's a delicate transition, and it is important to note that the visionaries driving this field are not advocating for the complete eradication of the traditional randomized controlled trial.
Not at all. For broad-spectrum pharmaceuticals like a novel daily statin for cholesterol or a new seasonal allergy medication where the potential patient pool number's in the millions and the risk of taking a placebo is negligible, the traditional RCT remains the absolute cleanest method for cutting through the noise.
So it's not one or the other.
Now, the consensus points toward a hybrid future.
The concept of the pragmatic trial illustrates that hybrid approach perfectly. Instead of the brutal 50-50 coin flip, researchers implement a 3-to-1 randomization ratio.
Right.
So 3 actual patients receive the experimental therapy for every 1 patient who receives the placebo. That immediately reduces the human toll of the trial.
But a 3-to-1 ratio might lack the statistical power required by the FDA.
Right, the math isn't strong enough.
So researchers take that single real-world placebo patient and top them up with 3 matched synthetic patients generated from the data.
It is an incredibly elegant compromise. We maintain the robust statistical power required to verify efficacy while drastically minimizing the number of actual human beings who are denied potentially life-saving treatment.
It is paving the way for an era of living evidence, where the rigid barrier separating everyday clinical care from cutting-edge research begins to dissolve entirely.
It is a profound paradigm shift. And as we conclude this deep dive, I want to bring the focus back to you, the listener. Think back to the scenario we opened with a parent and the 5-year-old child facing a neurodegenerative decline.
It really grounds the whole discussion.
It does, because the debate surrounding synthetic control arms transcends data efficiency and statistical modeling. It represents a moral mandate. If the technological infrastructure exists to conclusively prove a therapy's efficacy without withholding it from a declining patient, the ethical argument is that we are obligated to utilize it.
I agree completely. The evolution of the 21st century demands that our trial architectures become as precise and as inherently humane as the targeted therapeutics we are developing.
Exactly.
There will be statistical hurdles and probably regulatory missteps along the way, but the ultimate objective is an accelerated pathway to cures within a fundamentally more compassionate medical framework.
Which brings us to the ultimate takeaway from this research. If the prospect of accelerating cures resonates with you, engage with the shift. If you interact with patient advocacy organizations, inquire about their stance on synthetic controls. Support legislative and regulatory efforts that provide the FDA with the resources to safely audit and integrate real-world data into their approval pipelines.
It's going to take public support to move the needle.
It really is. But before we wrap up today, I want to leave you with a final, somewhat provocative thought to mull over, building directly on the mechanics of real-world data we've been discussing.
Oh, this is a good one.
We established that your routine electronic health records, your insurance claims, and even the biometric data from your wearable devices constitute the exact material used to build these digital twins.
Right, your data.
Which means simply by wearing your smartwatch on a morning run or by going in to complete your routine annual blood work, your digital exhaust is continually feeding the global medical ecosystem.
Whether you realize it or not.
Exactly. So without you ever setting foot inside a laboratory, could the mundane data of your everyday life be acting as the synthetic control arm that helps validate a cure for a rare disease on the other side of the world? Are we all, right now, unknowingly participating in the clinical trials of the future?
It's a powerful thought.
Thank you for your curiosity and for taking the time to join us on this deep dive. We will catch you next time.